All right, well, welcome everyone. My name's Jay Scott. I'm one of the co-executive directors of Alex's Lemonade Stand Foundation, and I want to thank you for joining us here today. We have an exciting lecture today. This is a lecture in the Alex's Lemonade virtual lecture series. I think this is either the 10th or the 11th lecture so far in the series. If you're enjoying these lectures, let other people know about them. Just before we get started, I want to talk about the next lecture, which is happening Monday, is going to be the many genomic roads to ALL, leukemia, presented by Charles Mulligan from St. Jude's. That should be another good one. Just some ground rules. If you have questions, you can type them in the chat and we will have them answered at the end of the lecture. If you have suggestions for other lecture topics, please put them in the survey that's going to be also posted into the chat. We're always looking for new ideas for topics and things to discuss on these. So today we have an exciting lecture. The title of it is Unsupervised Transfer Learning for Rare Disease Transcriptomics. It's going to be presented by Dr. Jaclyn Taroni. Dr. Taroni is the principal data scientist at the Childhood Cancer Data Lab of Alex's Lemonade Stand. Her team develops robust and reusable workflows to study pediatric cancer and trains pediatric cancer researchers to better analyze their own data. I'm lucky to call Jackie a friend. She is a lover of birds, and she's got a great lecture planned for us today. And with that, I will turn it over to Jackie.
Great. Thanks, Jay. Wonderful to be here today to talk, as the title suggests, about unsupervised transfer learning for rare disease transcriptomics. As Jay mentioned, I work at the CCDL, or the Childhood Cancer Data Lab, which is a part of Alex's Lemonade Stand Foundation, which means Jay is the co-executive director of the organization that I work for. So I'm very happy to be here, but there were a couple kind of suggested lecture titles that Jay rejected, that I first suggested. So the first one of these were, I thought neural networks were magic, and I was wrong. So if you were lucky enough to join us on Tuesday for Dr. Casey Green's lecture, who is the director of the Childhood Cancer Data Lab, you may remember that he said things like, neural networks aren't magic, deep learning isn't magic, don't romanticize deep neural networks. So what I'm going to talk to you about today really got started because unsupervised neural networks, we thought they were going to work, and we weren't 100% right about that. So I'll be talking about kind of applying unsupervised machine learning in the context of rare diseases and some trouble that we ran into. The second suggested lecture title was just this series of ends, which is either a really, really bad sequencing run, or it may just have to do with the keyboard that I happen to have in my second bedroom in my house. But this would have been a really bad lecture title, so thank you, Jay, for rejecting this one. That was a good call. The last one is, what do I do if my big data only gets so big? So what do I mean by that? So if I have high dimensional biomedical data, I'm studying a rare disease, it's assayed a rare disease, and I want to use things like unsupervised machine learning. Being in a rare disease space, I'm going to necessarily have a constraint on my sample size, and that can pose a particular set of challenges, which are very pertinent to studying pediatric cancer. So that's what I'm going to talk about today. Before we get into it, let's talk about what I mean by big data. So anytime I talk about big data in this talk, I'm going to be talking about genome-wide gene expression data, or transcriptomic data. So what I start out with is a gene by sample matrix, where in the case of humans, I am measuring tens of thousands of genes in whatever kind of set of samples that I'm interested in. And the thing about this data is it's noisy, but it's also structured. So we know individual genes are coordinated in their expression. And what that means is we can do things like dimensionality reduction, or unsupervised machine learning, or feature learning, or however you want to think about it, and go from this perhaps noisy matrix to hopefully kind of more coherent patterns. Now here, the samples are still columns. We have patterns or latent variables that we learn through our dimensionality reduction that we have as rows. And what this is essentially combinations of genes that we're measuring. So this is kind of technically motivated. It's very convenient to go from measuring tens of thousands of things to maybe a few hundred things. That can be easier to work with. The other thing is it's perhaps more robust to measure combinations of genes rather than kind of individual genes that might be subject to noise. But what I'm mostly concerned about, and what I'll mostly talk about today, is learning biologically meaningful patterns. If I want to use these techniques for biological discovery, ideally the patterns that I learn will be biologically meaningful and tell me about things like perhaps the presence or absence of a cell type that I really care about in a particular data set, or maybe it's a particular biological process that's altered over the course of treatment, or during disease progression. So again, our goal is biologically meaningful patterns, a useful representation of our input data. So this idea is not unique to transcriptomics. It's not even unique to biomedical data. If you caught Casey's talk on Tuesday, he talked quite a bit about kind of image processing. So here's an example that Casey would talk about kind of in earlier talks quite a bit, which this is a paper now from quite a few years ago, where folks took millions of still images from YouTube videos, trained what's called a deep sparse autoencoder, where an autoencoder is a type of unsupervised neural network that's generally trained to reconstruct its input. And then what these folks found is a useful representations of patterns that showed up in these images from YouTube. And one of them happened to be a cat's face. So again, this is something called an autoencoder. Casey Green's academic lab had some success applying autoencoders to transcriptome data, which I'll be talking about today. One such effort was from a former student in the lab, Dr. Jaclyn N, who trained an ensemble of denoising autoencoders on a compendium of Pseudomonas data that was publicly available. And in training that model and then looking at the patterns that were learned, or that output that was learned, and looking at the values of that pattern across the compendium, they were able to kind of uncover a subtle aspect of the phosphate starvation response in this bacterium. So this was really useful for shedding light on a particular biological process by simply, not simply, but putting together large collections of data were really useful for that. Another example from Casey's lab comes from a former student, Dr. Greg Way, who trained a variational autoencoder on the Cancer Genome Atlas PanCan data. And that model learned patterns that stratified melanoma tumors. So this is also something that might be interesting in the kind of rare disease space. We'd like to learn patterns that help us distinguish between different groups that we find of interest. So this is also a very useful representation for us to learn. So these are great. Can we just use this to study the disease that we care about? And the answer is it depends. So these are what we call kind of data intensive methods. So these methods would ideally like that we have measured things in many samples. So in this case, it would be ideal if we were measuring tens of thousands of genes and we had thousands of examples or patients or samples that we're learning from. So in Gia's case, the Pseudomonas data was about 1,000 samples in a bacterium. And if we think about the conditions from which kind of that collection of samples comes together, it's often things that are kind of adding a stimulus, some kind of perturbation where we expect a large variation in response between kind of our mock treatment and our stimulus, or there's something like a knockout where we might expect kind of a large effect on the gene expression patterns that we find. In the case of Greg's work, that was TCGA. That's over 10,000 samples, I believe, that were kind of collected as part of this concerted effort, and someone kind of put this together such that there was a compendium of thousands of patients in that particular case. So that worked well in those two scenarios, but when I first started kind of working on this, I was at Penn on a T32 through the rheumatology department. And the person in charge of the rheumatology department is someone named Dr. Peter Merkel, who's a world expert in ANCA-associated vasculitis. So ANCA-associated vasculitis is a rare systemic autoimmune disease that impacts the small and medium blood vessels in the body. And it is systemic. So publicly available data sets for this assay different tissues that were affected by the disease. And it's also rare. So that means that largely the data sets that were available were a lot smaller than the two examples that I just gave. And this kind of set me up for a particular set of challenges as we went to approach this. This is a specific example I'll talk about, but the lessons are really, I think, more general. And we'll get into that. So let's talk about using unsupervised machine learning in rare diseases using a series of toy examples. So these are made up situations and made up data sets that I'm going to talk you through, but they are based on different kind of problems that we might encounter. And the main idea here is that data sets from rare diseases are not always easily comparable. So let's imagine we have these two toy data sets. One is a collection of kidney biopsies that were collected as part of a multi-center consortium. In this particular case, perhaps the cohort is made up of multiple tissue-specific, highly kidney-specific diseases, as well as systemic diseases that happen to have kidney manifestations. And the disease that I, as a researcher, am interested in is only severe kind of diseases represented in that data set. Now here on the right is an example of a whole blood data set. Perhaps this was collected as part of an investigator-initiated trial. This takes place over 24 months, so it's a longitudinal study. And in this particular cohort, it's enriched for older patients, especially relative to the healthy controls that are available. So maybe my research question is, what pathways are perturbed in multiple tissues in this disease? And let's talk about training models on each of these and how this may work towards my question or not. So let's start with our kidney question. So, or data set rather. If I take an unsupervised machine learning model and I train it on my kidney data set, which again is a multi-center consortium, it has different diseases contained within it, what might I learn? So one thing I might learn is kidney cell type specific patterns, which is really biologically interesting, but it might not speak to my question about different pathways perturbed across the different tissues. It might learn something, essentially these patterns that are shared across different diseases that are in the collection of data, which again, very interesting, might not answer my question. And because this was collected in multiple places, I might learn something like a technical nuisance effect that I don't, isn't gonna help me for biological discovery, quite the opposite. And I might learn something about when and how the samples were processed. So this can be kind of a challenge to answer my research question. What about the blood data set? So this is, in this scenario, we have whole blood investigator-initiated trial. Let's say that in this case, the treatment under study is designed to selectively deplete a cell type, and we see that kind of as we go on, we see kind of a drop in the cell type represented in the orange here that we would expect to change throughout the course of treatment. And the composition of the sample is then going to change. But we know that cell types kind of interact with it and one another, you know, they're not in a vacuum. So what might happen, we might see kind of a depletion of another cell type represented in the light blue alongside it. So when we go to learn a pattern, maybe our model kind of has trouble parsing these two cell types, which can be a challenge. Ideally, we'd kind of be able to tell these apart and really dig in. Another thing we might learn from this particular data set is something like age. So ideally, we'd learn a pattern that was indicative of disease state, but if we have a cohort enriched for older controls, older patients relative to our controls, we might learn something about kind of blood composition dependent on age. So these are some challenges when we collect different data sets in different tissues. But let's say we train 2 models. We think we get something really interesting that speaks to this question of shared biology across diseases, and that's great. We still have some challenges ahead of us. So one of them is that when we have multiple models, we have to compare them. So in the case of the kidney data set, if I learn a bunch of kidney cell specific patterns, those might not readily translate, or we wouldn't expect them to readily translate to our blood data set. And this can actually take a lot of human analyst time. So it can take a lot of time to kind of dig into this and compare the 2 models. So what if we took a different approach entirely? What if we instead learned from a generic collection of biological data? We get useful representations. We then transfer that useful representation to our rare disease data set of interest. And this is an idea called feature representation transfer. And again, the idea is I have biological data. I can extract useful features that tell me that are biologically meaningful and tell me kind of about the biology in this collection, and then apply that to my rare disease of interest. And I gave just a moment ago an example of a multi-tissue kind of systemic disease, but it also is helpful when we have more comparable, arguably, cohorts where we're looking at a single tissue or single disease type. But even these can have their own collection biases, for example. And in this case, even if we fit models separately, it doesn't get around the problem of now we have multiple models and what do we do? So this is helpful in this case as well. So this is appealing. This is like, would be very useful as an analyst to be able to do this. What is it going to take for this to work? So what we need is for what we train on, the collection of data that we train on, and the collection of data that we want to go then transfer to be sufficiently related. It's helpful that a lot of biology is shared. And the other thing that's important is an appropriate model. We need a model that's going to learn a useful representation from a generic collection of data. So let's take the data piece for a second. So we're working with transcriptomic data. One of the great things about transcriptomic data is that there's a lot of it. There's a strong culture of data sharing, and what is publicly available, there are now millions of assays that are publicly available. It really represents a generic collection in the sense that it's whatever scientists decided to do experiments on, publish on, and then kind of release. So we have this kind of generic collection available for us to learn from. And I just have to quickly give a brief plug. The folks at the CCDL, we built a product called Refined.bio that you can go to and search for kind of biological data sets that are of interest to you. And we've uniformly processed this publicly available transcriptome data. So the URL for that is Refined.bio, and I encourage you to check it out. But back to the issue at hand, we have lots of data. It's whatever scientists thought were interesting. So we can then use it. But our goal is going to be the same. Our goal is still to get these biologically meaningful patterns, and we need a model to do that. And we can take advantage of the fact that individual genes are coordinated in their expression when we think about transferring. So let's talk about the model that we used. I was fortunate enough as a postdoc when I started thinking about these problems to go to a talk by Dr. Maria Cecchina, and she talked about a method that her group developed called PLIER, which stands for Pathway Level Information Extractor. So it's going to do what we're aiming to do on that last slide, where we have our collection of gene expression data, and we want a set of, in this case for PLIER, these are called latent variables, where it's these patterns that we have now on a per sample basis, and we want these to be biologically meaningful. So there's something, when you run PLIER, you also get the C matrix, and what's important to understand about this for the purpose of this talk is that what this is how genes in Y combine to get B, but during the training, PLIER is set up to have some preference for pathways and sparsity. So what do we mean by that? Part of training PLIER is providing the model with a set of input gene sets that represent kind of prior biological knowledge, sets of genes that either are, for example, indicative of a particular cell type, or can work together in a particular pathway. So some of the latent variables then that are learned by PLIER are going to be aligned with these input gene sets that represent kind of prior knowledge. The other thing is it's going to try to make it such that these latent variables are associated with a low number of gene sets that are hopefully related. So by parsing kind of different biological signals, you can think about it that way, we have an easier time kind of interpreting the output of the model. So this is handy for another reason, which is not every not every latent variable learned by the model will necessarily be aligned with the pathway. So it can do things like capture technical variants as well. And I'll show an example of it doing just that. So this is a very interesting model. I like this method a lot. It differs from the autoencoders in the sense that there is some constraint on the patterns that it learns, such that some of them are going to align with biologically meaningful patterns. Then we can use it for discovery. Okay, so we have a model that's going to help us learn these biologically meaningful patterns. Why don't we just use that on all of our rare disease data sets? So even though that's the case, that we have a model that can help us describe the biology, it doesn't sidestep the problems that I presented before. Namely, it doesn't help us with the fact that if we have multiple data sets, we still have to compare models between them. We have to fit models and then compare them. The other thing that can happen when we have smaller data sets is that the gene sets that are associated with a pattern learned by the model can be associated with different signals, like different cell types or things like maybe some kind of proliferative process, where because this is associated with kind of this mix of signal input pathways, it might be a little harder to interpret. So an ideal case is that we have kind of a set of latent variables that we learn where the latent variable is associated with kind of a small number of hopefully related gene sets, such that you can say, oh, this is a particular kind of lymphocyte latent variable. So that is another challenge when working with small data sets. Okay, so we have a model that has some really attractive properties. Can we apply it to collections of data that were not kind of collected together, that are kind of disparate data sets? So one of the first things we did was to build a compendium that was a single tissue and a single disease. So for this, we used lupus whole blood. So whole blood is going to be this mixture of cell types and adaptive immune cell types. And lupus is pretty heterogeneous, but it's not a rare disease. So there's enough publicly available data that we can put together a data set that is, 1,000 or so samples and train a PLIER model on it. But does the model learn things that we would expect or are useful to us? So to orient you, what we have here are columns are latent variables, and our rows are input data sets during training that were provided to PLIER, and then kind of a more saturated value here you can kind of think of as showing that the latent variable is associated with the pathway. So what comes out of this model are things that we would expect in lupus whole blood. So specifically immune cell types, like different subsets of T cells, B cells, monocytes, neutrophils. And what we would think of as kind of a classic lupus signal, type 1 interferon. So despite being across multiple data sets, we are able to learn kind of biologically meaningful signal. Furthermore, if we take the subset of latent variables and their expression values for things that do align with pathways, and then we kind of do PCA in that space, we can see that kind of the data set specific effects look more reduced, or they're not as pronounced as when you look at the subset of the B matrix that's not aligned with pathways. So maybe it's then partitioning kind of the biological signal away from the technical noise And if we're analyzing this space, we're gonna do a bit better. Okay, so that is a specific case where we're looking still at a compendia that's kind of uniform in the sense that it is the same disease and tissue. But that doesn't help us in the case where maybe we want to look across conditions or tissues, right? We still have some challenges there. So instead, for this particular project, our collection of data is from a resource from Jeff Leake's group called Recount2. What we ended up using was about 37,000 human RNA-seq samples that were uniformly processed with a pipeline called RailRNA. We trained a PLIER model. You get the Z matrix out here. The thing that's important to note here is we're going to essentially use this to calculate the expression of multiplier latent variables in rare disease data sets. So this is where the transfer piece comes in. And this altogether is what we call multiplier because it's multi-data set, multi-tissue, and also it's very important to have a cute name. All right, so does this work? Does this model, by using collections of generic data, have some useful properties? So we did an experiment where it basically subset the complete data set in a number of ways. We either took random samples such that we got training sets of different sample sizes, or we divided up the input based on biological context. So what we mean by that is something like just blood samples using the metadata or something like cell line. And what is on the y-axis for all of these graphs are a metric, which is the proportion of input pathways. So we give PLIER a set of input pathways, and then we can ask what proportion of those input pathways is significantly associated with at least one latent variable. So the way I think about this is that you're able to recover more biological patterns if you have a higher proportion of input pathways represented. So this is the experiment where we randomly took a selection of samples, and we can see that as the sample size of the data set gets bigger, we recover a larger proportion of the input pathways. On this particular graph where we're looking at biological context, these are now ordered by sample size such that the blood data set is the smallest, but because it's a mix of different cell types and tissue is going to be a mix of different cell types as well, we recover more pathways than we would, even though we have a larger collection of cell line data. So it's both probably sample size that's helpful and also kind of a mix of tissues when we do our training that can help us recover more of these input pathways. So not all of the latent variables are going to be aligned with pathways necessarily. So we can also take a look at the proportion of latent variables that are associated with at least one pathway. So we see as we get a bigger data set, this number decreases. So there are more latent variable, a higher proportion of latent variables not assigned to a pathway. That's another way to think about that. That kind of holds true over here in our biological context experiment. And we see less than a quarter of the full recount set multiplier are associated with input pathways. So there's one way to think about this where, well, you're just letting in more noise. Another kind of more optimistic way to think about this is perhaps that we are learning coordinated patterns of expression that are not yet supported by our input gene sets because we didn't supply them to the model, or maybe we don't know about it yet. So that is another way to think about it, but kind of digging into that is definitely a research problem. The other thing that's useful, or one thing that we want to do when we're analyzing a large collection of data, is to be able to parse kind of different signals that we might have a hard time parsing. So for an example, a couple examples, type 1 and type 2 interferon can kind of be hard to distinguish looking at expression data. Same thing for different cell types of the myeloid lineage. And then kind of this last example here is looking at different phases of the cell cycle. And a kind of more saturated color is better performance. And what to take away from this is as we increase the sample size, we're better able to separate out different pathways that we might have trouble doing so when we have a smaller data set. So that is useful for helping us analyze our data to be able to tell these things apart. So hopefully I've convinced you that using a large collection of data is helpful in the sense that we get kind of models with attractive properties. But let's go back to our rare disease data sets. So going back to our ANCA-associated vasculitis datasets, we have a set of nasal brushings. We have a kidney data set, and we have a peripheral blood data set, PBMCs. All of these are under 200 samples, and we have our multiplier model. What we're going to do to actually analyze that is take each of these data sets and put them in the multiplier latent space, get expression of or estimates of the multiplier latent variables for each of these data sets, such that latent variable 1 in our NARS data set is latent variable 1 in our kidney data set and latent variable 1 in our PBMC data. So this is attractive and appealing. Is it a good idea? So what we did to kind of dig into that part is look at things like counts or estimates of immune cell infiltrate. So we had the model that we trained that I showed you earlier that was on lupus whole blood data. And one of those data sets had information about neutrophil count. And if we take the neutrophil associated latent variable, the estimates for that for the model trained directly on the lupus whole blood data, we can see it's moderately correlated with neutrophil count. So this might be because neutrophils are terminally differentiated. It might be because we would have done a better job if we were looking at proportion rather than kind of count. Nevertheless, we have some kind of baseline for things that are trained directly on the data we are looking at. If we then put the whole blood, lupus whole blood compendium in the multiplier latent space, look at the values for the neutrophil associated latent variable that multiplier learned, and then look at the counts, we can see kind of similar performance, maybe even a little bit better, but it doesn't hurt us to do this transfer step. The other thing we can do is then look at solid tissues, right? So we're not limited to looking at whole blood. So we can take the neutrophil associated latent variable, we can compare it to a tool called MCP counter that was specifically designed to look at immune cell infiltrate in solid tissues. And we can see that the values for the neutrophil associated LV and the estimate from MCP counter are pretty well correlated. And the nice thing is this tool, PLIER, is not specifically designed to do this, but it does it well. And in the PLIER paper itself, they also demonstrated that quite nicely. Okay, so we can kind of learn about neutrophils in different tissues. A concern always when you're doing something like this transfer is that you're going to create an artifact by not training on that data directly. So one thing that we looked at is the kind of values for the multiplier LVs and those that the latent variables trained directly from the model, trained directly on the NARS data. And what we were able to find is the kind of estimates or expression values between the two models were pretty well correlated when you do a match and say, okay, this latent variable in the NARS model is most similar to this multiplier model. And that correlation appeared to be kind of particularly strong when the latent variable corresponded to some kind of biology or at least pathway input. And we can see even if there's kind of moderate correlation between the loadings or this kind of match between the latent variables of the different models, usually the kind of expression values, the B matrix, the pattern matrix that we're going to use for biological discovery tends to be a little bit stronger. So one thing I didn't mention is that the data that we trained multiplier on doesn't contain any ANCA-associated vasculitis data. So this model has never seen ANCA-associated vasculitis before, but it can still help us learn about it. So there is a latent variable if we looked at, called its number 599, if we took a look at the loadings, so what genes kind of combined to give us that latent variable, the ones that are heavily weighted or up at the top are things that are ANCA antigens. So things that antibodies come up against or rise to in ANCA-associated vasculitis. And the expression of these, this latent variable is in this particular data set, elevated in what the authors said was kind of a more severe group. So even though we haven't seen ANCA-associated vasculitis in training our model, we can learn things about it. And part of the reason why, or I think the reason why, is because what this actually is probably telling us about is kind of normal neutrophil granule development. So this is a normal process that kind of shows up in a specific way in our rare disease of interest. And by looking at a generic collection of data, we can still learn about this disease, even though we didn't train directly on it. The other thing we can do, which is kind of how we frame this in the beginning, is do kind of multi-tissue comparison. So looking at across all of our data sets for concordance in our latent variable values. So this is a latent variable that's associated with a macrophage input gene set. And generally speaking, what this is showing is that this, the expression level for each of these in each data set is higher kind of in more severe disease, which is interesting. This is not the only example of that, but it was one that was of particular interest to our colleagues who were disease experts. So that was helpful in this kind of systemic disease context, but it also, the multiplier kind of approach lets us analyze multiple cohorts quickly. So here are two medulloblastoma data sets that are different cohorts. Each of these have cell type labels, and by looking at the expression values, we can kind of check for concordance in patterns across different data sets quickly without training models separately and then doing that comparison, which is really helpful. So this came out about a year ago now in Cell Systems. It's available on GitHub. It's openly licensed. Our models and our data is openly licensed as well. Another fun thing about this is that you can see the stuff that didn't work. So I talked about kind of these nice examples. There's stuff that didn't work that's out there too, which I think is kind of fun. So this came out a year ago. Like I said, we are not the only folks to have this kind of idea. So I just wanted to highlight another piece of work called Deep Profile. And this is specifically focused on acute myeloid leukemia, where there are a decent number of publicly available assays. So what I mean by that is they are able to put together a training data set that is 6,500 samples. But the problem here is that the labels are missing. So what I mean by that is maybe we don't necessarily have the phenotypic data that we're really interested in these kind of historical data sets. In this case, they were interested particularly in treatment labels, which this collection of data for the most part didn't have. So what they did was train a variational autoencoder on this collection of AML data without labels, took the low dimensional representation from that, took a data set that did have labels, transferred the low dimensional representation to that, and then used the values from that as input to predict drug response. It seems to do a bit better than training on kind of all of the feature space, so all of the genes. So that is one way to think about it. In a case where the tissue and disease, we have a large collection, but often we don't have the phenotypic data that we really need to kind of dig in or we want to dig in. But we can still leverage that and transfer it to a data set we really care about. One quick thing I wanted to mention, we started at the beginning. Jay mentioned that part of what we do at the CCDL is training workshops for pediatric cancer researchers where we show them how to analyze their own data. One of these we put on in March of 2019. in Houston. And one of the folks that joined us is Dr. Robert Alloway from Sage Bionetworks. He came, and when we were going over the section where we kind of worked through different people's own data, we kind of took a look at MultiPLIER together and started working with it. And I'm happy to say that kind of this set into motion a project that was published earlier this year with the folks at Sage taking the lead. And what they were able to show is that pathway-associated MultiPLIER latent variables are essentially, you can take them and distinguish or look at different tumor types in the disease of interest, which in this case is NF1. So that's interesting. Another cool thing that came from that is that, again, like less than a quarter of the latent variables learned by MultiPLIER have kind of annotations, if you will, but there are still unannotated latent variables that have expression that turns out to be associated with specific genetic variants. So that's one way that you can think about kind of digging in further. So even though things aren't aligned with a particular pathway at the outset, doesn't mean they're not interesting. So I just wanted to share this kind of story of someone applying this to their own data to learn about their area of interest. So just to summarize, hopefully you have been convinced that learning from a generic collection of biological data is appealing, and it's an appealing approach for studying rare diseases. Data availability is a challenge, so we can have limited sample sizes in domains like this. In some cases, we might have a lot of samples, but we're missing kind of desirable phenotypic data. And features can be shared between the collection that we train on and a rare disease. I talked about an approach specifically called MultiPLIER. That's an unsupervised transfer learning approach for transcriptomic data. We found that large varied collections of data allowed us to kind of extract more biological signal, have more things associated with our input pathways. And then we also, it was very helpful to be able to transfer knowledge from a single model to then use that and compare multiple rare disease data sets. So with that, I want to say thank you to the wonderful folks at the Childhood Cancer Data Lab that I get to work with every day. This project started while I was still at Penn. Peter Merkel was our ANCA-associated vasculitis expert. We worked with folks at the NIH and Michigan as well. And the last story I highlighted was really led by the folks at Sage Bionetworks. And with that, that is all I had. I wanted to let you know that you can visit the Childhood Cancer Data Lab at ccdatalab.org. This is our mascot for a refined bio product called Tubi. And here, once you get your data set, he is whisked away in a hot air balloon. So with that, I'm going to stop sharing.
Well, thank you, Jackie.
Thank you.
I thought that was really interesting. And if you are ready, we have questions that I'm going to start reading. This first question came in pretty early, and it's from Itamar. Biologically relevant or meaningful, who decides or defines this?
So I think it depends. So, which is not a satisfying answer, but it kind of depends at the scale that you're looking for. So it can be perhaps as quote unquote simple as being able to stratify different, in the example, like melanoma from anything else. But it could be digging into kind of specific subsets. That's biologically meaningful. I think it depends on your question, but ultimately we don't want to learn about things like what kit was used to prep the samples. We don't want to learn about kind of when the samples were run. But it depends on the context that you're trying to apply to, whether you find something, I would say, useful.
I just lost the questions here. Hold on. We had two questions come in with similar topic. Can MultiPLIER be adapted to analyze other omics data such as DNA methylation data sets? And then the similar question was, can MultiPLIER be used in other omics data besides transcriptomics? And if yes, how is it performed?
So this specific work that I presented is just for transcriptomic data, but I think some of the ideas can certainly be applied elsewhere. So I think if you are, again, able to learn a biologically meaningful representation from your methylation data, so things like cell type come to mind again, perhaps if you learn from a large collection of that, you are still able to kind of transfer that to a smaller data set. So the model that is kind of publicly available and that we put together is specific to transcriptomics, but I see ways that the idea could be potentially used elsewhere.
Okay. One person said, thank you very much. Next person says, thanks for a great talk. Do you believe the prior knowledge about pathways was particularly important for the effectiveness of MultiPLIER? And if someone wanted to apply a similar approach to some other kind of omics data, should establishing some kind of preference for latent variables be a priority?
So I would say yes, from my own kind of personal experience. I think that's very kind of useful. And I think it does have some definite benefits during training to kind of have that constraint. So yes, I would say that would be something I would look out for. I think it is helpful, but maybe not necessary. I would encourage people to check out the PLIER preprint as well. Kind of the first posting of that digs into that a bit. So I'd encourage folks to check that out.
Okay, Milton, his question is, autoencoders are able to find more complex patterns, nonlinear, but PLIER allows to incorporate prior knowledge, as you explained, and it's easier to interpret since it's a linear combination of gene expressions. Do you know if there is a deep learning model that combines the advantages of PLIER, easy interpretation, et cetera?
So there may be at this point, nothing is coming to mind. I know that people have kind of done things where they, in the supervised setting, where they do impose some constraints on the neural network, and they can use that to analyze their data. So it is probably possible to kind of put similar constraints. I know people have done it in a supervised setting, but that's definitely an area of interest to then kind of leverage the kind of advantage of this interpretation. So I think there are ways to do it. It's a open research question, I would say.
Okay. So Arthur Frank asks, with supervised learning, how do you deal with small data sets, as in the case of rare diseases? Do you augment your data set? And if so, how do you do that?
So yeah, in some cases you can do something like kind of bootstrapping to augment your data set, if you will. One thing that I'm kind of interested in, which is not the question that was asked, is if the things that this low dimensional space can help us in the supervised setting, in that we take, like the deep profile example that I showed, if we take the low dimensional representation and use that as an input to a predictor in the supervised kind of setting, if that would help us. So that's a little bit of a different question than what was asked, but it is something that I think is interesting.
All right, I think you're going to like the next one, Jackie. Robert Alloway asked, with the growing bolus of public single-cell data, do you anticipate building a single-cell multiplier?
Sounds fun. Yeah, great. Let's do it. Maybe Robert can do it. Yeah.
Hendrik says, thanks for answering his question, for the talk. Jeremy asked, if you started this project today, would you use recount2 or refine.bio for training MultiPLIER?
So I would say that one thing that's kind of interesting by getting larger collections of data, Refined.bio is bigger than the set that we used for this particular project that was from Recount 2. I'm kind of curious if you get diminishing returns, where like getting a bigger data set doesn't help you learn anything quote unquote useful anymore. So I think one area that I am interested to dig into is using a Refined.bio compendium and kind of doing similar experiments where we kind of start to understand whether or not we kind of can't learn anything useful anymore by increasing our sample size. The other thing I'll say is that Refined.bio has microarray data in it. I looked at kind of training some of these kinds of models in zebrafish briefly, and I do think kind of the increased sample size of kind of adding that different platform is useful.
So you think you'll try it? Maybe.
Yeah.
Okay. Ashish says, have you guys tried using integrating multi-omics data sets and then use the latent matrix Z for transfer learning? And then it says using MOFA tool.
The short answer to that question is no, we have not tried that, but that would be interesting. Yeah. I haven't gotten there yet.
Okay. Now this must be a follow-up to a previous question by Itamar. It says, do you compare to supervised results?
So I think maybe the framing between, there's a kind of distinction from what we were setting out to accomplish by using MultiPLIER in this setting, which was kind of to get these patterns and kind of in an unsupervised manner find out what's in those rare disease data sets. So, which is a little bit different from, say, trying to predict response to treatment, which I would think of as that is a supervised machine learning problem or framing.
Okay. Last question, Jackie. It comes from Iwen, and the question is, compared to the traditional analysis that uses significant genes to perform gene set enrichment assay, do you think MultiPLIER is more capable to pick up key driving pathways or molecules for disease pathogenesis?
So I think the using a matrix factorization approach like PLIER, you're going to fit your data a little bit better. And another thing that I think is really helpful is that it is unsupervised. So in the sense that you do not have to know your groups ahead of time. And that can be really powerful to kind of parse things apart. So I can see kind of scenarios where gene set enrichment analysis is going to kind of tell you what you need to know, that kind of the rankings of the gene, you're able to kind of distinguish some kind of driving factor. And, you know, certainly I've seen things like that in analyzing different data sets in that manner. But I do think one of the kind of powerful parts of using an approach like this is not having to define those groups ahead of time. And that can be really useful. That's useful certainly in a case like ANCA-associated vasculitis, where the manifestations are different, and setting up kind of a two-group comparison, especially if, for example, you don't have controls, that can be a little tricky. So I would say that there are some things that we kind of like about this particular approach, in that we don't need those labels or groups ahead of time.
Thank you, Jackie.
Thank you.
That was the last question, but I would feel bad if I didn't send a shout out to Joan, who was in the house.
Oh.
So welcome, Joan. It's nice to see you attending. And thank you so much, Jackie, for a great talk. There was a lot of interest. We had a lot of questions. And interestingly, the number of attendees grew throughout the talk. So I think people were sending messages to their friends to join in. I've never seen that happen before in any of our previous 10 talks. So thank you, Jackie. You did a great job. And thank you, everybody, for attending.
Yeah, thanks everyone so much for coming out.
Yeah, if you have suggestions for future talks, fill out that survey. And with that, we will end this one, and we'll see you on the next one, everybody. And
Thank
have
you.
a great weekend, Jackie.